I will call to order this meeting of the Oversight Committee. It is now 8:30 a.m. Dr. Hernandez, please proceed with the roll call.
Yes, good morning. Dr. Cummings,
Present.
Mr. Payne,
Here.
Mr. Margot,
Here.
Mr. Montgomery,
Here and in compliance.
and Dr. Patel,
Yeah.
Dr. Rice, Dr. Rosenfeld,
Here.
Mr. Taylor. We have quorum.
Very well. Thank you very much. Just as a point of order, I would like to have the Oversight Committee aware of the weather constraints that we have today with staff and with OC members and travel. So we would ask that your questions and comments be germane to the presentations that are made, and any additional questions or comments can be made to the staff at a later time. Thank you for your understanding. We have our draft minutes from November 20th Oversight Committee meeting in your agenda packet behind tab 1. Are there any corrections to the minutes as circulated? The chair will entertain a motion to approve the minutes of the November 20th Oversight Committee meeting.
Second.
All in favor, vote aye.
Aye.
Any opposed? Motion carries. Moving to item 4. Our next agenda item is public comment. Texans created CPRIT. Opening our meetings with public comment underscores this board's commitment to transparency and accountability. Mr. Ellis, has CPRIT received any requests to provide public comment?
We have not, Mr. Chairman.
Very well. Moving on to item number 5. The chair now recognizes Dr. Michelle Lewan to introduce Dr. Laura Indolfi, who will give a grantee presentation.
Morning.
You need to move it towards you. Thank you.
Incredible blend of scientific innovation and entrepreneurial expertise to the field of oncology. And I saw you just met Laura in person, and her bio is actually printed under tab 2 in your booklet. So I will save everybody's time. Here's Laura.
Nice meeting you. Thanks for having us presenting Panther. Do you need to say something else, or can I?
Yeah,
Where are the slides?
they're putting the slides on.
Okay. So in the process, while they're waiting, while we wait for the slide to be queued up, thank you very much for first of all awarding us with a CPRIT grant over 2 years ago, but also for all of the support and for inviting us to present the progress that we have made since we got the grant award. It has been a very fast 2 years, and I feel like at home today, like coming from Boston, it feels like I'm back in time where we started with the snow and the cold weather. So trying to get a sense of where the slides will show up.
Well, we are finding Dr. Indolfi's slides. I may resume some of her introduction. So she's the co-founder and CEO of Panther Therapeutics, which is a clinical stage company focused on developing localized cancer treatment solutions. So their proprietary platform, Sagittarius, is a drug delivering platform that targeted to bring continuous and targeted cancer therapy without systemic side effect. So the drug PTM-101 is already in early clinical stage for treatment of pancreatic cancer. Dr. Indolfi's contribution to the field has earned her significant recognitions. She's named TED Fellow, an honor that awarded only to 21 global changemakers annually. She was also invited to Fortune Most Powerful Women Next Gen Summit. Additionally, she was selected as one of the 5 distinguished women for the inaugural 2018 Mass Next Gen program, recognizing her exceptional leadership in biotechnology. She holds a PhD in biomaterials and both MS and BS degrees in material science and engineering from the University of Naples, Federico II in Italy.
And if there are no slides, I can go with the printouts, but I don't know if it is needed for the video.
So for
Just
the
give
committee
us-
members, the slides actually is behind her bio page.
Yeah, we're behind tab 2. We'll give our IT team just one more minute to see if we can get the slides on the screen.
So they are slide. I'm happy to. I don't know if they want to put the video, but yeah, yeah. But yeah, but I'm happy to do it.
Apologies, Dr. Ndolfi. I guess we will go ahead. Just we'll follow along in our
Yeah,
books
then.
here. And if we can get the slides, they'll catch up with us.
Perfect. So as I mentioned, we relocated to from Boston to Austin in 2022. We have been setting up operation here since then and expanded our clinical development as well. So as you can see on what is labeled slide 3, I can walk you around on what is the company vision and what we are trying to do. So we are all very familiar with the side effect that cancer treatment affect patients when they undergo their regimen. And that is because even though there has been 40 years of development in new molecules and in new drugs to be designed, the way we give the drug to the patients hasn't really drastically changed. Patients either get an infusion through the bloodstream or do they get pills that allow the drug to go everywhere in the body with only 1 to 5% on average really reaching the specific organ that the therapy is intended to treat, poisoning the patients in an attempt to cure the cancer. And that leads to the nausea, the vomiting, all of the very ill side effects that patients experience that most often than not contributes and lead to the death and the mortality of the patients. So what we are coming to with a different lenses is having an engineering approach to a medical problem. And we know that there are drugs out there that have been effective. We are finding a way to bring them directly where they are needed and to leave them there for a prolonged period of time so that they don't get washed out right away, but they can stay in proximity of the tumor and have an effect in the long run. And on the next slide, you can see how we are engineering them. We are leveraging the new innovation in cancer diagnostic that is allowing us to reach organs deep into the body. There is minimal invasive procedures, there is robotics, there are a lot of tools that right now are becoming the pillar of cancer care. Here we are. Perfect, thank you. That are becoming the pillar of cancer care, but currently used only for diagnosis purpose, to take a biopsy or to have a camera to look around. And we are designing product that can be delivered through those tools so that we can put them really in proximity of the tumor and not having open surgery. Coming from a cardiovascular background, that's where we got the inspiration, where we went from open heart surgery to minimal invasive procedure to stent, balloons, and now even the valve can be placed with a little catheter through the leg. So we used all that we have learned in those 40 years and translated into the interventional oncology and moving on our innovation. Skipping back to the slide. And sometimes, pictures, are worth 1,000 words. So I've been speaking for 3 minutes here, trying to tell you what we are doing but here is a visual analysis of what we have. On the bottom side of the slide, you can see what happened when a drug is delivered through the bloodstream, and we use fluorescently labeled drug in green. You can see that on the bottom left, that is a depiction of the liver. All of the drug, majority of the drugs gets there, that the tumor is so macroscopically fluorescent that it has a halo around it. While on the bottom right, that is a picture of the tumor, it's pitch black. There is nothing, there is no much of the drug that actually reached there. And that's what we are trying to innovate and solve. And as you can see on the top of the slide, on the right, now we have the tumor that is macroscopically fluorescent, is homogeneously depicted in green, and the liver, as well as any of the other organs that they are usually affected by the drugs, they are pitch black. There is no drug that escaped the place where we place our product, and enhancing us to have high efficacy with very minimal side effect. At the foundation of Panther, there is a technology that is really a broad platform. We spun out of MIT with several IP portfolio that have been fully licensed by the company. And right now we have 2 products in development. We have a peritumoral treatment that what we are bringing into the clinic with pancreatic cancer is a biodegradable film that we have engineered to be unidirectional delivery so that we can control precisely the geography or the anatomy of where the drug is distributed. And we have also an intratumoral treatment to be placed directly within the tumor core through the same minimal invasive procedure so that we can have a dual effect. But most of the presentation today is going to be on the peritumoral treatment, as this was the effort of the CPRIT Award, for which we have started and focusing on pancreatic cancer as our lead indication, even though once we have clinical proof that this technology has a meaningful clinical benefit, we can expand in other type of indication as well. So talking briefly of what we are treating for pancreatic, what was our approach for pancreatic cancer and why we chose to start there from a company and from a clinical perspective. There is a huge unmet clinical need, even though there has been several work done in the last 10 to 15 years, the overall survival is still 13%. That means that only 13 of the patients that will get diagnosed this year will be still alive 5 years from now. And that is a number that, even though has been improving in the last 5 years from 8 to 13, is still nowhere close to the 80 or 90% that could be possible for breast cancer, for example. So the unmet need is huge and is there. There is also a good commercial opportunity from the company perspective to be viable, even though the patients, they are not as broadly vast like in lung cancer or another type of indication. Then there are several reasons, when we tell people that our first product is for the treatment of pancreatic cancer, we get some of the comments of, are you crazy? Pancreatic cancer is such a deadly disease, like nothing has really worked. So I take a minute of your time to tell you why we are not exactly crazy. We have been thinking about that. We have a reason why we keep moving pancreatic forward in our development. And so let me share you, we may be crazy, we are not naive. So let's put it in that way. So pancreatic cancer are poorly vascularized. That means that they are sitting in the middle of the abdomen. They have very few blood vessels that actually go to the tumor. And so this idea of delivering drug through the bloodstream and changing the drug, but having the same route of administration, it's really full game. Like nothing is going to change unless you change the delivery route. And so that was the perfect cancer for us to start developing so that we could prove that the route of administration and our treatment in proximity of the tumor is what makes the difference. From a clinical perspective, there is a bias that pancreatic cancer patients are only diagnosed when they are metastatic. And that is true and false at the same time. They actually only 50% of the patients are diagnosed metastatic. That is in line with some other tumor type. But because, as I mentioned before, systemic is not working, then they will progress very fast to metastatic. And so the span between non-metastatic and metastatic and then slippery slope to be very sick, it's very short. And the ability to prevent that transition, the ability to control the tumor burden and ideally reducing it so that the patients can go to surgery has a huge clinical benefit, even though it doesn't require the tumor to fully disappear. And so again, a localized sustained treatment was the perfect tool for us to prove that that was an ideal approach. So for all of these reasons, every time we kind of evaluate the pipeline, we keep putting pancreatic cancer on the front because it will really give us a meaningful effect to change the life of those patients without the need to have the burden of curing them or making the tumor fully disappear. And if there are questions, please, interrupt because I appreciate the interactive. So this is a video that hopefully will work. Yes, of what our first product is about is as a drug-eluting layer and then has a backbone drug-free. It's flexible that it can go through the laparoscopic trocar that are used for minimal invasive procedure that are currently implemented on those patients. And the vision is to provide surgeon extra tools that they can use during the surgery after they've visualized the area to leave it behind and serve as a cage of the tumor so that it cannot continue progress and expanding. The layer with the drug will degrade first, allowing for the drug to go only towards the tumor site, enhancing the reduction in tumor volume. And then the entire product will degrade so that we do not need to have a multiple procedure and we don't need to have a procedure to remove it at the end that the drug is being released. So the same way that patients right now, they can go every other week to get an infusion into the clinic, we envision they could go every 2 to 3 months to have a product placed. It's in outpatients procedure. They don't need to be hospitalized overnight, and they come in the morning, get out in the afternoon, and have within them an implant that can deliver a high dose of the drug for 30 days or more. So that has been the vision that we built Panther on and the development we have been moving forward. This I already touched upon in the sense that the metastatic patients, thinking ahead of how we were starting the clinical trial, metastatic patients are the 50% of the population. The other 50% is a spectrum of localized tumor, but not necessarily operable because of different reasons. Either they can touch some of the vital organs that are surrounding the pancreas, or because there are vasculature involved that will make bleeding a very difficult procedure for those patients. So this is the patient population we have been focusing on, and we have been advising, and we have been thrilled to have been recognized the possibility to enter into the clinic as first-line treatment. That is something that in cancer is usually very hard to get to and very thought about. Usually when you have a new innovation, you enter into the clinic as third or fourth-line treatment. The patient has already failed several prior treatment, and they're already sick by the time that they get a new drug. What the innovation we are bringing to the table allowed us to get clearance from different regulatory agencies, both in Australia and here in the US, to enter at the beginning of the treatment, like first line, or at least first treatment as the patients are newly diagnosed. They will receive our procedure because it's minimally invasive, and then they can go on and have their standard treatment. So we are not interfering with any of the further treatment that the patients will have. We are augmenting the response that they could have, again, without adding toxicity overall. We started with the first trial in Australia. We treated 3 patients at our lowest dose. We have demonstrated that we can easily integrate into the clinical paradigm. Those patients were diagnosed, set up for laparoscopic placement, went on their standard treatment. There have been no side effect additive due to our placement. There has been extremely safe, no infection, no inflammation due to the procedure. What was really interesting too is that we had no hematologic toxicity. So we are giving the equivalent, in this study, we gave the equivalent of 1 dose of chemotherapy that is given systemically into the product. And usually when you do that with the drug that we have selected, you start having nausea, vomiting, air loss, neutropenia. We didn't see any of those side effects until they started other treatments systemically. And this is because we have been able to demonstrate that we do not have drug into the bloodstream, like the picture I showed at the beginning. Like we place the product in proximity of the tumor and the drug stays where it is, because what we have done is we took one of the pitfall of systemic. There is no blood vessel that get into the tumor. So everything that you go through the blood vessel doesn't get to the tumor. That also means that everything you give to the tumor cannot escape anywhere else because there is no vehicle for that. So we are forcing the drug to stay where it's needed. It doesn't go anywhere. It doesn't have side effects. And we have been very pleased to show that in all of the 3 patients we treated, we had an anti-tumoral response, even at our lowest dose, that was intended to show only safety of the procedure. So let me walk a little bit about what those data look like. So as a reminder, and I will have something to pass along so you can play with, because show and tell is always a good thing. So we manufacture these right now at different dose. They're intended to be into the body, and into a cold day like this, they tend to be a bit stiff. So I've been warming it up in my hand a bit. But this is what the product looks like at different dose. We place it minimal invasive on top of the organ on the area underlying the tumor, like depicted on the left side of the graph. And what happened is that as the product degrades, the drug elutes in one direction. So we are showering the tumor basically from the top to the bottom with this flow of drug. And what we have been able to show is that the tumor actually reduces size into that direction preferentially. So start at this different, at this specific area. And then if we monitor over time the dimension of the tumor, we can see that they deflate like, souffle that they went wrong. They started at the size and then they start reducing it in volume. That is something that physicians have been receiving interestingly, because even in the few cases where pancreatic cancer respond to systemic treatment, usually you don't see a preferential route of reduction. So that was a validation that our technology was having an effect. And then we asked ourselves, as an engineer, that's great. It's very cool. They're reducing size. We proved the technology. We have demonstrated what we wanted to design. But does that has any effect for the patients? Are we doing better for the patients that they will be doing systemically without our product? And so we compared the overall volume reduction, the overall mass of how much it reduced versus what is expected as a reduction for clinical study and real world data when they have only neoadjuvant treatment, when they have only standard systemic treatment. And on regular, like from a study from MD Anderson of over the 300 patients, a median volume reduction, so expected when there is good treatment from neoadjuvant, from systemic drug, is around a 20% in reduction. That is not everybody, but if you average between the one that they respond and the one that they grow exponentially, usually they're expecting a 20% in volume reduction after 12 weeks. And what we were able to demonstrate is that we can show a 60, a 34%, and that will continue over time. So we are adding to the powerful potential of the drug by being able to have a first punch up front that is delivering the drug only locally. And as you can see, the bigger drop in response is within the 12 weeks. So where we do have our product on top of systemic. So this first study really helped us understanding that our technology was working. We were able to develop what we were designing for, but also we were having at a dose that we expected to be only safe and not effective, also an incremental and significant additional effect in tumor response compared to what it was given from systemic treatment. So with this data, we went back to the FDA and we applied with CPRIT right after we were in middle actually of that study. We didn't have all of those results yet. But the next couple of slides, they are more about what we have achieved since you granted us the word. And as you can see, there has been quite a bit of progress in different areas, but let me walk you quickly on each of them. So as I mentioned, we moved from Boston, we expanded our operation from Boston. So Now we opened an office in Big Cave in May of 2023. We have now an operational lab within that premises that allow us to do lamination process and post-production characterization of all of this manufacturing material that we have. It is quite of a bit, is overall 8,000 square foot of lab space between office, R&D, and manufacturing clean room for potential clinical production. So we really started, put our foot here, ready to expand. We actually, there is a lot of echo in some of those areas, like there are few people than space, but we really did it for the intention to expand and grow and have a presence here in the long run. We then made an evolution of the manufacturing process. So as I finish talking, I can pass this around. There is no, just a piece of plastic. It's a cool day. But so at the beginning, when we were doing our first human study, we were making this one by one. We had individual manufacturing, the people were in the lab doing one at a time. We could do a maximum of 20 per product, per batch size. So it was good for our first human study, but then we needed to scale up. For this new study over the last 2 years, we have improved the manufacturing process. Right now, we have a continuous film that sheets that we can manufacture, allow us to have 120 products made from the same batch solution so that we were able to supply and manufacture enough for this upcoming clinical study. And we have also further improved the manufacturing to be automated towards registrational study and to support commercial manufacturing as well. So the road for there is still a couple of years away, but we have started that process of manufacturing, and we definitely have been able to manufacture enough of product for this clinical study. The team has completed the clinical GMP manufacturing campaign. We have a study that is upcoming with double and quadruple the dose than what we tested in our Australian. So we had done clinical lots at both of those doses. We have right now in total 690 products that were manufactured over the span of less than a year. We use some of them for characterization, stability, and further improvement, and some of them have been shipped to our facility for then deployment to the clinical site. See if this video will work. Yes, so this is a different dose. So it's a thicker dose. You have seen a little bit what they feel like, prepared to be ready to get implanted. They can be rolled extremely tight so that they can fit into the trocar for the placement. And they maintain their structure. They have not cracked. There is no this deficiency into the product itself. And that is true across the doses, from the 100 milligram that you have seen passing around to the 400 milligrams that we have developed. And then, as I mentioned, we have tons of those products. We have manufactured them. They are a third-party logistic partner ready to be shipped to clinic as we are ready for starting the clinical trial. We have also, during the last 2 years, finalized and complete all of our IND-enabling animal study. We tested over 36 pigs, 3 different doses, the lowest, the highest, and an exaggerated dose to make sure that safety was going to be taken under consideration. And we were able to fully characterize release profile of the drug and toxicity once implanted. With all of those data and all of the CMC and manufacturing data we have done in parallel, we submitted an IND last summer, and we got cleared in full during the 30-period stage review. So we had engaged with the agency along the way. We had several pre-IND meetings. We have engaged with them to get feedback ahead of this submission so that we will come prepared and be ready to move into the clinic smoothly. And indeed, that's how we have been executing on that. Right now, the clinical trial is fully approved. All of the documents are FDA approved. We have a central IRB approval as well that allow us to add more sites if needed in a very smooth way. And we have partnered with all of the clinical vendors that they are necessary to make a trial a success. So for lab analysis, for central imaging, for CT scan, as well as the full CRO that will support the study running. The clinical trial is up and running on clinicaltrial.gov. We are, we have been engaged with PanCAN, the Pancreatic Cancer Network Association, to have the study being included in their database as patients start to get diagnosed, enriched, and do their research so that they are aware of what we are doing. They are engaged with our clinical development plan, and they will be able to help us identify any patients if needed that can be beneficial, that can benefit for our approach. We are ready to dose our first patients. We have 5 sites selected, one of which is MD Anderson. That was a longstanding collaborator of us since the inception. 3 sites are expected to begin enrollment in March. We have spanned them throughout the states so that we can kind of have a good pool of patients that they can come from different large center that there are center of excellence for pancreatic cancer. The study we will be starting is up to 30 patients, so we may not need a lot of study to run, but we plan for success, but also have contingency. So we have another 5 sites that we are continuing the process of identifying to make sure that if needed, we will have an additional pool of patients to supplement the clinical study. We ran our first one in the midst of COVID, so we never know what's going to happen and how clinical operation can be affected. We got scarred by that, so we are trying to be prepared. So for any additional delay, we will have a backup plan. And this is a picture of what our clean room looks like right now in our Bee Cave facility. It was the former facility of Apollo Endosurgery. So they already had a clean room set up for medical device. We have scaled up the design for upgraded them to a drug device combination settings. We have all of the drawings in place, and we will plan to start construction ideally by next year as we get more source of funding to execute on the ability. And that facility will be able to support both our registrational study, but also early commercial. So we will give us a good presence to start with and then continue growing from there. Takes a village to tell what I said very fast, and I'm very proud of the village of superhero that have been supporting us and staying with me throughout the process. They come from different backgrounds because we are a combination product, and we need to have really different set of skills and expertise to make sure we have a handle of all of the different moving parts that we have. We have a small team, but they are very mighty, so I'm really proud to be able to grow them together with the company as we develop. And all of that work allowed us also to be featured on Times Square. That is something that as a Italian immigrant entrepreneur, when I moved here in the US, that was definitely not where I thought one of my projects was going to be featured. So I am glad to them for all of they have allowed us to accomplish, especially being able to bring a product to the clinic and make a meaningful impact for patients. So that's all. Hopefully I have been
well
time.
done. Thank you so
you.
much, Dr. Indolfi. This is fantastic presentation. And as a medical oncologist, I can tell you that, the groan down the hall when we get a new patient of pancreatic cancer, especially, in the locally advanced stages, we just have so little that we can really offer them. And this is a new technology that is very exciting. So wish you the best and look forward to more updates. I'll open it up for questions from my colleagues. Dr. Rosenfeld.
Excellent presentation. Thank you for being here. Did you say that your initial 3 patients were done those studies were done in Australia?
Yes.
Could you explain why they were done in Australia?
Yes. So the data in Australia, like clinical data for phase 1, are accepted by FDA for translation into further development. He allowed us to have a faster and more capital efficient development plan. We were able to start clinical trial with the GLP with the tox and the acute preclinical data that we had at 1 dose, while FDA will have asked, as they did, to test all of the dose and above. So we were able to enter into the clinic and start collecting clinical data and clinical proof of concept as we were continue developing and collecting all of the data needed for FDA and IND submission. So it's been a dual path that was allowing us to better understand the product and the process, but also being able to use those data for further clinical development in the US. And in fact, that when we submitted the IND, the FDA agreed, based on the strength of the data we have provided from Australia, to let us start at the 2nd dose directly and didn't need to repeat the original study. So from a company perspective, it was a capital efficient way to gain time that is very important when we have limited capital.
No question? Yeah.
Dr. Hernandez?
Good morning. Great presentation.
Thank you.
Did you say you're in Texas, or where are you located?
In Austin, I'm
In
here.
Austin. Okay,
Yes.
good. All right. Let me make sure you were in Texas, not lost in New York somewhere.
No,
Oh.
I am fully here.
Great. Now the mechanism, I just want to make sure. So it sounds like it's dose and proximity dependent. So if you lay it on top and the tumors, obviously, whatever shape it is, the closer it is, you said it starts dying off. I know you're imaging it. Do you check for viability of, how do you know that the dose on the top will eventually make it to the bottom? Because you get scar tissue, and do you check like PET scans? I'm just curious how you do it.
We have been debating that quite a bit in how to do it in the patients. We have done it in the animals, like, because we can take the tumor over different time and controlling penetration, and we do have a scar formation that it gets.
I'm sure.
In the patients, we initially tried to have biopsy collected over time so that we could have sample of the patient of the tumor back. Pancreas is a very sensitive organ, so having a lot of it was not approved by the IRB committee at the FDA. There was pushback due to the unbalanced risk benefit of booking the patients for a biopsy versus the need. So for now, we are doing it with the imaging only.
Just volume.
Volume and CT scan.
Regardless, just volume. You can't measure cellular activity with a PET scan or MRI, right? You can't do that.
No, so in our next study, we do have a PET MRI included.
Great.
Not all the sites do PET MRI for pancreatic cancer.
Yes.
The technique is not as consistent and as reliable that they feel they can get meaningful data. So it's a site-to-site specific. If they do have into their protocol the ability to do PET MRI, that is included into our protocol, but we have CT as the main to be consistent across sites, so that we have at least one site of imaging that is the same across all of the sites.
I think cellular activity is important because then you're going to mislead people, right? Your tumor may shrink.
Yeah.
But the bottom or the center part may be still alive because it's heterogeneous. It won't get there.
Yeah.
And so you're giving this false perception to people, like let's say a patient, that you're getting better when in reality you still have that root cause still being present. That's the only reason I was asking.
Yeah, and it is, I agree, like, but also what we are trying to, what we see this technology really to have an impact is if we can reduce, we're not saying we are going to cure the tumor. It's not like it will go everywhere. It's improving compared to what you get systemically, but because it's local, we could clean some of the area that could make the patients eligible for resection.
Yes.
So even if the core is still active, but they can go to surgery, it's a balance. But we have been trying to implement different methods to have activity within the cells as well.
If I may,
Dr. Patel.
I just say it in Italian, grazie. I have 2 questions, very quick questions. The first one is as regards the you have a bio layer of your drug and it elutes in a unidirectional way. Are you considering multiple layers where you'll include biologics and also radiation sensitizers? It's very important to do so. I was just wondering.
Yes. So the way we are manufacturing is by layer manufacturing. And we are envisioning a way where we can have different layer with different compound. Regarding to the radiosensitizer paclitaxel, that is the drug we are using it for our first one. It is a radiosensitizer that has been selected because in conversation with physicians, they will really appreciate the possibility to have high dose of paclitaxel in proximity of the tumor when they do radiation. We needed to choose how to move to the clinic fast to be able to have a proof of concept. So our first product is fully into the recycling business, let me put it this way. We have a drug that is known, we have a polymer that is known, and we are really trying to show that the innovation is into the technology. But the next step is, right now we have a very well-established layer by layer manufacturing. Is gonna be very smooth and easy and straightforward to transition to have different layer with different compounds that you can put in together. And linking to the previous comment, we could put something that is disruptive of the tumor microenvironment or the scar, and then enhance penetration and enhance response of any immunologic immunotherapy. So.
And the second question is that, you had done the initial phase 1 studies while understanding fully well it's a phase 1 study, and that survival was increased by 7 months. In those 7 months, did you look at quality of life? Because given pancreatic cancer, the way it is. Thank you.
Yes, we did. And we have very few patients to really make a difference, make a statement, but we did had some intriguing results from that questionnaire from quality of life. So we have integrated that into this study that is upcoming, and we will continue collecting the data set to make sure that we will gather information on quality of life as well.
Dr. Rice.
I'll just make one comment. First of all, thank you so much for the presentation, the very exciting progress that you're making. I wanted to just observe for the medical students and those interested in a healthcare career, maybe physician career, maybe oncologic career, that those of us that are old enough to remember the 1980s when interventional cardiology was being born with things like balloons, as you mentioned, and interventional radiology has come along in ensuing decades, interventional neurology, and I think interventional oncology may be something that will explode in the coming decades with the technology that you're describing. And so I wanted to speak to the medical students out there wondering what's interesting to do in their medical careers, and would say interventional oncology might be a very early but exciting arena for them to work in.
Yeah, we are on the cusp of really starting to see what interventional oncology will become, and the same like interventional oncology and all of the interventional cardiology and all of the other. 20 years from now, I think that we will be in a completely different place where hopefully we will not need to see, loved one dying because of kidney failure because of the drug they were taking when they had a tumor somewhere else. So.
Excellent points. Thank you so much for your presentation.
Thank you very much to CPRIT for the support. Like nothing of that will have been done in the timeline that we have done it, and in the space we have done without your support. So thank you very much for believing in us.
You bet. Thank you for being here. All right, let's move on to item 6. This is the Chief Executive Officer report behind tab 3. We'll ask Ms. Doyle to provide the Chief Executive Officer's report.
While I'm getting myself set up, I will say, Laura is here in town, so she and I will go out to lunch sometimes. And she told me after she attended JP Morgan in January, they had brought together women that are involved in life science and in biopharmaceuticals. And she told me something, that there are more CEOs named Michael of of biopharmaceutical companies than there are female CEOs of biopharmaceutical companies. So it was just a very interesting factoid. And of course, you know that all of our awards are based solely on merit, but it is exciting to have, to be supporting a female-led biopharmaceutical company. So we're thrilled for this. So members, my report is behind tab 3. Dr. Indolfi's presentation is a perfect way to start my CEO report to you. As you may remember, CPRIT statute requires the CEO to report on the progress and continued merit of each research program annually. So you'll find my report beginning at page 350 in your meeting book. With the grants that you will consider today, CPRIT has awarded nearly 2,100 grants to worthy cancer research and prevention projects throughout Texas. Like Panther Therapeutics, these projects are each making progress every day in our audacious mission to prevent, detect, treat, and survive cancer in Texas. Our progress is measured in the cancer and cancer precursors we have detected early, the future cancer cases forever prevented through vaccinations and education, new discoveries every day that fuel the next early detection method, the clinical trials that make novel treatments available to the sickest patients now, and the drugs, devices, diagnostics, and services that are available on the market to detect and treat cancer. In short, the lives that have been saved and extended through the work done in Texas, funded by CPRIT and approved by you. So we time the annual CEO report for the meeting that follows the publication of CPRIT's annual report, and this is no accident. The 2024 report is a showcase of the work that CPRIT grantees are doing and the progress that is being made right now in Texas. I would like to recognize everyone on the CPRIT staff for their contribution to this year's report, which is based around the theme of clinical trials. This is truly a collective effort by our grantees and by the CPRIT program staff. But in particular, I would like to acknowledge the efforts of Mark Leffler, Justin Rand, Bridget Barstow, Shannon Kuzik, and Royce Hart. Their contributions to the annual report are significant and behind the scenes. And this is even, it was a bigger lift this year because it came right on the tail of publication of the 2024 Texas Cancer Plan. And that same team was very involved in the presentation and the writing of the document of the Texas Cancer Plan that you see. For the rest of my report, I'll quickly go through some of the things that have developed since I submitted the memo for this packet. One of the things that I'll point out is that we have 53 full-time employees. That includes 9 long-term employees. The reason why I point that out is because we are above our current FTE cap. And so we now report that to the Oversight Committee every quarter and then also to state leadership. And since our last meeting in November, of course, I've updated you this through in our activities reports that are also in your meeting book. But the draft budget came out, House Bill 1 and Senate Bill 1. And in both of those proposed bills, they include CPRIT's requested 10 additional FTEs. So we're very thankful and supportive of that. That will help us continue our mission. So I'll close out with a brief legislative update. Like I said, since my memo in your meeting book, the Speaker of the House, Dustin Burrows, has named 30 standing committees. Representative Dr. Greg Bonin has again been named chair of House Appropriations Committee, with Representative Mary Gonzalez of El Paso as the vice chair. Representative Gary Van Dever is the new chair of Public Health, and Representative Liz Campos is the vice chair. So CPRIT will present our budget request to the House Appropriations Committee this coming Monday, February 24th. Dr. Rice will represent the Oversight Committee at the House Appropriations Committee on Monday. Cindy Payne did the same for this for the Oversight Committee and for CPRIT at the Senate Finance Committee last month. I'm very grateful for the Oversight Committee support, and it sends a powerful message to the legislature and state leadership. I'll close out with the number of bills that have been filed as of yesterday. We have 4,854 bills that have been filed for consideration this session. So it will be both a marathon and a sprint to get to June 2nd. And I'll answer any questions.
Mr. Margot.
There'll be between 6,000 and 7,000 bills filed
Yeah, we should all,
between,
I
by the end of, before,
This- we
before
should put a
March.
bet on that, but maybe,
Yeah,
maybe I shouldn't
so-
be saying that. Yes, I mean, I think we had a record last session. I think we'll have a record this session. Do you remember how many bills were filed during your session?
It was always around the same, as I recall,
Yeah.
close to 6,000, around 6,000 something.
Yeah.
I mean, several people think that's notches in their belt when they file bills.
Any other questions for Ms. Doyle?
Just a real
Mr.
quick one. With the additional employees that could come on board, is there enough room in the office to accommodate those people?
The short answer to that is no, there's not. But luckily we have a very robust remote work policy, and that helps us manage both our in-person location and then all of our staff.
Mr. Taylor?
I did notice that it was filed yesterday. Yes. Yeah.
Sorry. It was filed yesterday and all it says in the headlines is cancer clinical studies. Do you know what Senator Parker's goal is?
Yes, this is actually a bill that that Senator Parker passed when he was a representative. So this is the Cancer Clinical Trials Participation chapter. So CPRIT has issued 2 RFAs based on that legislative authority. So the bill that he has filed, and I saw that it was filed yesterday. I haven't had a chance to study it, but it helps clarify some of the language in that chapter.
Very good. Thank you so much, Ms. Doyle. We will now move on to item number 7. It's our pleasure and duty to recognize Mr. Burgess to present the Chief Compliance Officer's report.
All right. Just one time. Well, it's good to be back. And Mr. Margot, I just want you to know, I did get the flowers and chocolates you sent. So,
It's your favorite.
right. Yes. And also, I do see a few I Love Compliance pins today and it makes my heart happy. And today's your lucky day. You get 1 million compliance bucks for that. And I don't know what you'll do with those, but I'm sure they'll come in handy one day. My update is behind tab 4. And I'm just going to highlight a few pieces from the compliance program update. CPRIT has approximately 1.5 billion in grants under management and about 540 active grants at any one time. And again, that varies from quarter to quarter as some grants close out and we award new ones. And we receive about 540 reports monthly, and we help monitor those on a weekly basis. As of the end of January, we had 19 reports that were outstanding. And so I think that is a good number. If there is a good number, it's under our 5% kind of self-imposed threshold. So we are doing well. And again, we meet weekly as a staff. Most of CPRIT staff attend that meeting and monitor those on a weekly basis. In terms of financial status report reviews, my team, we review those. They come into the agency. They're reviewed by grant accountants, and they come to the compliance team for a second level review. And we reviewed about 533 of those reports this past November, December, and January. And 80 of those, or 15%, required a resubmission, inaccurate, missing documentation or inaccurate information. So we typically reach back out to the grantee, get that information, and those are resubmitted. We performed 8 enhanced desk reviews in the November, December, January, and 9 on-site reviews. And those are a little more extensive. Of course, we go on-site. We did offer a virtual option over the past few years, and we're slowly sort of retiring that policy. So most of ours are on-site, and it's kind of exciting in the sense that, my staff go out and they get to see firsthand and talk with the grantees, and they see their enthusiasm. They see they tour some of the facilities, and it's a really good experience when staff come back and talk about that experience. You may recall last year about this time, we had quite an influx of match expenditure reviews from product development grantees. So as you know, they're required to have a match for their grant, and we review 100% of their match expenditures. And already this fiscal year, we've reviewed a little over $33 million in grant expenditures. But for context, I believe it was in FY22, we reviewed just under $15 million. So that is exponential increase in the amount that we're reviewing. The good news is that I believe last year it was just around 29 million, and we identified about $320,000 worth of unallowable match expenses. So far this year, with a 33 million, we've identified about $1,000. And so somebody do the math on that. That's 0.0003% or something. So, and I attribute this to the great work that we do as a staff, the trainings that we put on for our staff. And one of the things that we have started doing this past fiscal year is for our product development grantees, we're taking that a step further. So as a new grantee, you come in, you get a new grantee training, but also prior to submitting your first quarter financial status report, we meet with you, we come alongside you and provide a little extra layer of technical assistance. We look at the expenditures you're submitting, we look at the format that you're submitting them in, and we kind of help walk you through that. And so we feel like that's having a tremendous impact on the ways that grantees are sort of adhering to our standards. And again, for many of these grantees, it's their first foray into state funds and receiving. And as you know, with state money, there's a lot of strings attached to that. And so it's not easy, but we do try to help them to manage that. So those are some highlights from my report. I'm happy to answer any questions.
Thank you so much, Mr. Burgess. Do we have any questions? Mr. Margot?
The 10 FTEs that you added, are they, Kristen referred to, are they in your area, Vince?
One.
Just one?
Yes.
Are you staffed enough to do what you need to do?
This is a trick question.
An open-ended.
But possibly, potentially, mostly. And in terms of like the match reviews that I talked about, most likely we're going to have to go to a sampling method in the future. We won't be able to maintain that level of review as we continue to award product development grantees in increasing numbers, or create a system where if you have a 100% clean review for 2 years in a row, then you get a year off or you're sampling or something. So we'll have to monitor that closely. But I definitely have the support of Ms. Doyle and the leadership to, but as you know, to add staff, it would be for us beyond this legislative session. It's an ask 2 years from now. So, but we'll manage.
Thank you so much.
Thank you for your
report.
concern, Mr. Margot. I
Yes.
appreciate that. I know you are. Thank you.
We'll move on to our compliance certification. Turning to the certification of the award slates that we will consider today, Mr. Burgess provided the written report certifying that the proposed academic research awards and review process complied with CPRIT statute and administrative rules. Are there any questions regarding the certification from Mr. Burgess? Hearing none, we're going to move on to, thank you so much for your reports.
Absolutely.
Great to
Thank
see
you, sir. Thank you.
We're going to move on to item number 8. This is going to be behind tab 5 in your meeting book and behind starting on page 4 of the awards packet. The chair recognizes Dr. LeBeau to provide the academic research program update, and following her update, she will introduce the program integration committee's grant award recommendations.
And these are for fiscal year 24, review cycle 2, fiscal year 25, review cycle 1, and recruitment cycles for fiscal year 25, cycles 2 and 3. I mentioned these were 53 awards, and they total $67.8 million, and they are displayed in table 1 on this slide and on page 4 in your proposed grant award book. If we go to the next slide, and the next one, please. Thank you. The priorities addressed for the academic research program are shown here. And 46 of the awards address a broad range of innovative investigator-initiated research projects. But I'm very pleased that we have 6 recruitment awards, and a number of our awards also address important priorities such as hepatocellular cancer, childhood and adolescent cancers, and computational oncology. If we go to the next slide, I'll refer you to pages 6 through 9 of your proposed grant award book to see the slate of individual investigator research awards. There are 28 recommendations. I think that if you peruse these pages, I think you'll see that this is an excellent mix of basic and translational studies. The grants address important questions in cancer biology and cancer metabolism, drug resistance, metastasis, studies to improve immunotherapies, and mitigating therapeutic toxicities. Next slide, please. The next slate for individual investigator research awards for cancer in children and adolescents. And there are 6 awards that are recommended here, and they are shown on pages 9 and 10 of your booklet. I'm pleased that of the 6 awards, 4 of them are focusing on brain tumors and 1 is focusing on neuroblastoma. And these are really critical tumors in children. If we move to the next slate, this is the slate for computational systems biology of cancer, shown on page 11. There are 3 awards recommended, and I think these awards are proposing exciting approaches using AI and systems biology to dissect mechanisms of drug resistance and therapy selection. The next slate on table 6 is the Individual Investigator Awards for clinical trials. And there are 3 awards that are recommended, including an award proposing combination therapy for metastatic breast cancer and an award proposing microbiota transplants. This is a really hot field right now. This award is proposing transplants in the context of immunotherapy, but in this case, it's to reverse the disruption of the microbiome that has been associated with antibiotic treatment. The last slate of individual investigator awards is shown on this slide, and this is on the next 2 slides, actually. And these are the Prevention and Early Detection Awards. And there are 5 awards that have been proposed, and they include studies of blood-based biomarkers for early detection of recurrence in head and neck cancer and smoking cessation grants. With your permission, I'll just keep going through the slate and you can, we can reserve questions. The next, okay, we'll go one more, please. Thank you. The next award shown on table 8 is a single competitive award for the Collaborative Action Center. And this center seeks to position Texas as a national leader in reversing the trajectory of hepatocellular cancer. And they have a number of goals. The first is to provide research support and promote connections between researchers through a new population research core. Their second goal is to inform public health officials about hepatocellular cancer and influence public policy to reduce the burden in Texas. And their last major goal is to support education of healthcare providers and the public about hepatocellular cancer. And this is through a number of mechanisms, including an annual meeting and the creation of educational materials and standard operating practices for preventing hepatocellular cancer. The next award shown on the next slide is a Core Facility Award. And this is a cancer agent target discovery and aptamer development core. And this will serve researchers across Texas in identifying targets for intervention in cancer and developing cancer therapies using aptamer technology or oligonucleotides and peptide molecules to bind to specific targets. The remainder of my awards are recruitment awards, and I will describe 6 to you. The first is a recruitment of an established investigator who has been nominated by Rice University, Dr. Pernilla Wittung-Staffshed, and she's been nominated for an appointment as a professor in the Department of Chemistry at Rice University. And she is a established and really renowned scientist in inorganic chemistry and biophysical chemistry. And she specializes in studying metal dysregulation and has actually pioneered a new research field around the role of metal ions in folding and function of metalloproteins. She focuses specifically on copper, which is often a cofactor in essential enzymes, and plays an important role in a number of cellular functions, including in cancer, it plays a role in migration and metastasis. So recently, she has identified a number of new copper transport proteins and pathways for copper binding proteins. And what she is proposing to do is to characterize and identify the copper-dependent pathways that promote breast cancer cell migration and metastasis, and to use this as a starting point for new approaches to prevent cancer metastasis. I think one of the important aspects of this work is that you that it should extend to many types of cancer, as well as provide new prognostic and predictive biomarkers. The remaining slate of 5 candidates that I'll present represent first-time tenure-track faculty members. And the first candidate is Dr. Daniel Gao, who is being recruited to Baylor College of Medicine, to the Department of Biochemistry and Molecular Pharmacology. Dr. Gao has an outstanding record in protein engineering, genome biology, and genome engineering. And in particular, he's developed a new technology for genome editing. And this editing method allows the integration of large DNA sequences into the human genome. So it's similar to the CRISPR-Cas9 technology. He proposes to use it for 2 main projects. The first is to use it to edit the human genome so that he can study the functional significance of variants that have been identified in cancer genes like BRCA1, and in particular, variants that are called variants of unknown significance, where we can't predict what their function will be on the protein. So he wants to alter them and then test biologically what that function is. His second project is also an important one, and he wants to be able to use this technology to enable the rapid development of CAR T cells by rapidly inserting large DNA modules into CAR T cells, with the hope that this will advance the method of preparation of CAR T cells for immunotherapy. Collectively, these 2 approaches may advance cancer prognosis and precision treatments in clinical settings. The next candidate is Dr. Rosalie Griffin, who is being recruited to MD Anderson Cancer Center. She's a human computational geneticist and will join the Department of Epidemiology. Her research combines state-of-the-art genomics and computational techniques to inform risk stratification, early detection, and interception in lymphoid malignancies. She will be mentored by Dr. Christopher Flowers, who is a CPRIT Scholar. And Dr. Flowers has developed a very remarkable resource, a very large and diverse prospective cohort of lymphoma survivors. And she will use this database and tissue base to study a phenomenon called clonal hematopoiesis. And she will study large mosaic chromosomal alterations and determine what is the role of these genomic abnormalities in risk for developing lymphoid malignancies, and to determine how this can be used for early detection and interception. The third candidate is Dr. Nipun Verma, who is being recruited to Baylor College of Medicine. And Dr. Verma is a physician scientist, and in particular, a radiation oncologist. And she has a very unique multidisciplinary background with extensive experience in molecular and cell biology and cancer immunology. She has conducted during her residency program research using a CRISPR screen to look for targets that impact the killing of cells by CD8 T cells following radiation treatment. And she identified a number of genes which, when altered, impact the response to radiation and immune therapy via interaction with the CGAS STING pathway. And she will take advantage of these findings to evaluate some of these interacting proteins, one of which is expressed on the cell membranes after radiation treatment and suppresses the immune response. So she has developed an antibody that she wants to use to prevent the function of this protein, to prevent it from suppressing the immune response following treatment. And with this, she aims to improve the immune response to immunotherapy. The next candidate is Dr. Tao Zhu, who is being recruited to University of Texas Southwestern Medical Center. And Dr. Zhu is a superbly trained physician scientist who has a strong background in tumor immunology and lung cancer. And in particular, he has been studying cellular RNA processing and modification pathways in lung cancer. And what he has determined is that if you block the metabolism of cellular RNA within tumor cells, it can cause those cells to activate the innate immune sensor pathway. And in lung cancer cells, when the cells activate this pathway, it can cause cell death. So what he is proposing to do is to target the innate, to use this approach to target, to activate the immune pathways and really in lung cancer cells and really effectively trick them into mounting an antiviral immune response that blocks their cell growth. And I think this is a really interesting novel approach to harness the antiviral immune machinery, but may also be useful for other types of cancer as well. The last candidate is Dr. Ziwen Zhang, who is being nominated by the University of Texas at Dallas, and for appointment in the Department of Chemistry and Biochemistry. He has world-class training in chemical biology and drug discovery, and he's discovered a novel methodology with engineered antibodies to reprogram the cellular process of autophagy. And as you'll recall, autophagy is a process that cells use to break down and recycle harmful contents within the cell, such as unwanted proteins and damaged cell components. And what he thinks that he can do with these functional antibodies that he develops is to fine-tune the autophagy response by using antibodies that are directed towards some of the key targets of autophagy, protein aggregates and dysfunctional organelles. And in doing so, he thinks he can regulate and activate autophagy in a controlled way, and thereby induce cell death instead of inducing the unwanted off-target side effects that some of the other drugs that target autophagy have seen in the clinic. So that completes my description of the slate of 53 awards. I'm happy to address any questions you may have.
Thank you very much for the presentation of this slate. It's robust and exciting, the work that is due to be done. Do we have any questions? Dr. Hernandez.
In reference to your collaborative action program for the hepatocellular, the one educational, what is the Baylor College of Medicine, what is their portion they're putting in for this endeavor they're doing? Because this is a lot of education. This is a lot of what the medical school does already through the family residency, GI, internal medicine to educate the public. And so I'm just wondering, what are they putting in?
So they have a very large component. So it includes interacting with community organizations to hold events for educating the public. They've developed materials in multiple languages that describe risk for hepatocellular cancer. They have been working with the Texas Department of Public Health and the DSHS to develop a hepatitis C elimination program in Texas and a screening program. They have worked with a number of hospital networks to integrate cancer hepatocellular screening into their routine SOPs. So I think it's really through a multi.
A lot of the things you
approach.
described, my point is they're supposed to be doing it already. And I'm just wondering why I mean, it's done, but I'd rather use this money to recruit another scientist to Texas or product development, because what you're describing
what-
is their charge to do already as medical.
Right, right. So one of the things that the CAP program allows them to do is just to disseminate these approaches throughout Texas. So they have been very effective in not only organizing their own programs, but in disseminating them and sharing them with other groups, teaching other groups how to do this.
To collaborate, how to work
Yeah.
together. Okay.
Any other questions? Dr. Rice?
And I was just going to extend on that. It seems to me when that was first done, there was a lot of energy to sort of get all of the best thinking around hepatocellular cancer in Texas organized in such a way that there would be sort of leveraging of knowledge and leveraging of strategies. I don't know if that's
Yes.
how you would
Yes.
describe it, but wondered if you could go down that track a bit.
Yes, they've actually been very effective in doing that. The award has also supported pilot projects, and they have also been able to coordinate. We've also awarded a number of individual investigator research awards that focus on hepatocellular cancer. And these investigators are all part of this consortium and participate in the activities. They have established a very large database and biobank of patients with cirrhosis and MASLD, which has been used by a number of investigators. So they have over 300 publications now from investigators that are part of this consortium, part of the hepatocellular consortium. And that includes a number of improved methods for early detection on blood-based assays using a panel of, they now have a panel of methylated DNA biomarkers as well as metabolites in blood that can be used to identify patients at risk.
Just to extend one more second, again, being sensitive to time, you mentioned the data set, the large data set they have in a biobank. Those sounds like, does that have value? And I'm wondering if you could give us a comment on sort of the context of the impact of CPRIT and the return on investment, to use a terrible term in this context, but wondered if you think a biobank has significant value potentially over the next few years or decades.
I think that this is an incredibly unique resource that will that will foster a number of studies that cannot be done anywhere else, because you need those tissues, you need that clinical data, you need that population of patients at risk. So for example, I think the return on investment has been outstanding because it has enabled a number of investigators to apply for additional grants which will be based on samples from the biobank and clinical data from the biobank. The group at Baylor in the last year received a program project award from the National Cancer Institute to expand their work on identifying risk markers for patients with hepatocellular cancer. So I think it has been a great return on investment. And now that you brought it up, I think this would be a great group to bring to talk to the Oversight Committee and talk about some of their accomplishments.
Let me just, again, one more second, that biobank, and I apologize, here we go, but that includes not only genomic characterizations, but proteomic characterizations and metabolomic characterizations, right? So that there's sort of a full stack of science that has yet to be even worked through.
Yes. That's
And
right.
that sort of future value is actually astonishingly important, correct? Would you agree?
I agree with you entirely. And it's just like having a library. If you don't have books, nobody can ever do any studies. So now there's a whole battery of different types of studies that can be proposed once you have this biobank and the materials stored.
Thank you.
Mr.
Thank you for your.
Chair, is
goodness.
it possible to get a list or a compendium of studies that are now possible that were not possible without that biobank? That would in effect answer Dr. Rice's question about how what we're doing being productive in novel areas. Am I clear? Yeah. I think we're going to have a discussion on that later. That information would be extremely helpful. Here's what we're doing that no one else can do. And here's what we're making available that no one else can. Thank you.
Good point. All right. So with no further questions for Dr. LeBeau, we'll move on to the compliance certification and conflict of interest notifications. I'm sorry, Dr. LeBeau, have you finished your presentation?
Done.
Thank you. Mr. Burgess has certified compliance for the academic research award process. It is my understanding that Dr. Rosenfeld has reported a conflict of interest with one of the application we will consider today. Are there any other conflicts of interest that have not been reported? Hearing none, members, you have the list of applications and grant amounts recommended by the Program Integration Committee for the Academic Research Grant Awards. We will approve the PIC's recommendation if 2/3 of the Oversight Committee members present and voting agree. There are 9 academic research award slates constituting 53 grant recommendations. We'll take a separate vote on RP 250278, which is the application that Dr. Rosenfeld identified as having a conflict of interest. For the remaining 52 recommendations, rather than taking a separate vote on each grant mechanism, I will ask for a vote to approve all of the slates. If a member wants to consider one or more award recommendations separately from the vote on all the slates, please make a motion to do so now. First, we will consider RP 250278. I will entertain a motion to approve the PIC's award recommendation for RP 250278. All in favor, vote aye. Any opposed? Hearing approval from at least 2/3 of the members present and voting, the motion carries. I will note for the record that Dr. Rosenfeld did not vote on this recommendation. I will now entertain a motion to approve the PIC's remaining 52 recommendations for the following academic research grant award mechanisms: Collaborative Action Program to Reduce Liver Cancer Mortality in Texas, Collaborative Action Center, Core Facility Support Awards, Individual Investigator Research Awards, Individual Investigator Research Awards for Cancer in Children and Adolescents, Individual Investigator Research Awards for Computational and Systems Biology of Cancer, Individual Investigator Research Awards for Clinical Trials, Individual Investigator Research Awards for Prevention and Early Detection, recruitment of established investigators, and recruitment of first-time tenure-track faculty members. All in favor, vote aye. Any opposed? Hearing approval from at least 2/3 of the members present and voting, the motion carries. I will now entertain a motion delegating contract negotiation authority to the CEO and CPRIT staff to authorize the CEO to sign the contracts on behalf of CPRIT. All in favor, vote aye. Aye. Any opposed? Motion carries. Thank you, Dr. LeBeau. I'll now move to item 9, which is behind tab 6 in your meeting book. The chair recognizes Ms. Magid to provide the prevention program update. Good morning.
Good morning, everyone. It's a brief update. We're currently in FY 2025 review cycle 2. Peer review actually takes place starting tomorrow. And we hope to be able to present some recommendations to the PIC and to the Oversight Committee at the May meeting. And for FY 2026 review cycle 1, we will plan to release 3 RFAs: the primary prevention of cancer, cancer screening and early detection, and the dissemination of CPRIT-funded cancer control interventions. The RFAs will actually be released on Friday, and applications will begin being accepted starting March 19th. We will also conduct a funding opportunity webinar for interested and potential applicants, and that will be held in mid-March. And that is the end of my update, and happy to answer any questions.
Thank you, Ms. Magid. Are there any questions regarding the prevention program update? Very good. We look forward to the next meeting. Thank you.
Thank you.
Let's move to item 10, the Chief Product Development Officer report. This will be behind tab 7 in your meeting booklet. The chair recognizes Dr. Michelle Lewan to provide the Chief Production Development Officer's report. As a part of the update, she will ask our approval for the proposed FY 2026 product development RFAs. Welcome.
Thank you. Good morning. So my update will have three parts. First is overview of our 25.1 cycle award. We received 90 applications, and among those, 32 of them are from out of state and from 12 different states and 3 different countries. 24 of those applications are invited back to submit a full application, and among which you approved in last November 9 to be awarded. Among those 9, 5 of them are out of state from 3 different states and also a foreign country, Germany. And among the 5 of the awards are full company awards. One of them is a device and diagnostic award, which is given to a San Diego company, Curve Bioscience. Since the announcement of the award, they have already certified their matching fund and relocated to Dallas. So start working on their goals and objectives. It's a diagnostic blood test for liver cancer. And also so if you look at our full company award, it seems like 4 therapeutics and 1 diagnostic, but our 3 seed award actually came each from a different mechanism. Although we do not fine tune in our RFAs, but the 3 companies are 1 therapeutic, 1 device and diagnostic, and the third one is a new technology. So actually the application pool and our selection process has been very successful, as you've seen in last November. Okay. Second part of my update is the new RFA that we are going to release and open the application portal next Monday. It's a supplemental award for 25.2, and we have total available funding of 10.3 million to be awarded with the cap with awards capped at 3 million or 30% of the current award. So eligibility will be current awardees with an active PDR award awarded before 24.2, which means the companies will have at least 1 year to develop their project towards their goals and objectives and show significant progress. So the rationale being we want to help companies, and especially the companies that already awarded, their project went through vigorous review and screening and also due diligence process. But because of the environmental change, sometimes inflation, price increase, other times interaction with FDA, so they have to do some adjustments to their studies and price increase, etc. So some of our awardees, although making good progress, but sometimes the research will be delayed due to other factors, and a little bit cash help can help them bridge the gap and make bigger and faster progress towards their goals. So that's why we are releasing this awarding mechanism. And the timeline is printed on the screen, and review will be interview will be conducted in April. So because they are already our grantees, so the applications will be reviewed by our PDRC and led by the lead reviewer who have been reviewing progress report for those awardees and / applicants. Hopefully we are targeting to report our award recommendations in May. Lastly is the action item I need your approval for. It's our RFA for 26.1, and we have 4 funding mechanisms, 3 full company award funding mechanisms. They're therapeutics, device and diagnostics, and new technologies. And those awards are not capped for 3 years in duration. And then the fourth one is a seed award. Seed award campaign includes any type of research like therapeutics, device diagnostics, and new technologies. The award is capped at $3 million and also for 3 years. That concludes my update. Thank you.
Thank you very much for your presentation, Dr. Lawan. Do we have any questions? Yeah, I had the same question. Dr. Hernandez asked, what does that really mean when you say the first 3 awards are uncapped, that they can award any amount of money?
They can request any reasonable amount that justified by their plan of research. And our reviewers are yes, so they, in their application, they're gonna submit an itemized budget. So our reviewers is gonna look at the budget and decide whether the application is, the asked amount is reasonable.
Very good. Thank you. Okay, any other comment or presentation? Thank you. The chair will entertain a motion to approve the proposed FY 2026 product development request for applications. All in favor, vote aye. Any opposed? The motion carries. We're going to now move to agency business, item 11 behind tab 8 in your meeting book. The chair recognizes Mr. Graves from Weaver and Tidwell to present the internal auditor report.
Good morning. My report to you is fairly brief. We are really in the middle of all of our internal audits right now. On page 8.1 of your handouts, you'll see our 3 big audits in our audit plan for this year is post-award grant monitoring, a procurement and P-cards advisory engagement, and then a non-grant expenditures audit. We started the post-award grant monitoring and the non-grant expenditures together because they really center around some of the same folks and a lot of the same processes with the disbursement of funds. We started those on January 13th. We actually just on the 14th of February had our planning discussion with all the personnel in procurement and P-cards over that advisory engagement. We'll get it kicked off later this month, next week on the 24th. And so we are really just in the middle of everything. Everything's going very well. We expect to have everything wrapped up and be ready to report to you by the next Oversight Committee meeting. The other 2 areas we have are communications and IT general controls follow-ups. Those are slated for later in the spring. And that's just due to the timing that it was needed to elapse so that those issues could be addressed by the CPRIT team. The only other thing I'd just call to your attention on page 8.4 of your handouts is our open item summary. So the series of tables preceding that are just the internal audit history. And you'll see that aside from the general controls follow-up, which we gray out due to sensitivity IT issues, we only have 1 open item really to follow up on, and that's from the communications. It's an IT-based finding, and we expect it to be remediated this year. With that, I can stop for any questions.
Thank you very much, Mr. Graves. So at the very end, I think you said the open finding that's listed here from 2018, we're going to be able to close that out this year.
I anticipate so. We
Okay.
expected it to be last year. That finding centers around a tool on the website that allows for translation into Spanish,
Right.
and some other accessibility issues. And that tool has been selected and tried to be implemented and then reselected over the last several years. So that's what that finding is.
Got
It's
it.
really
Thank
about
you.
website accessibility.
Got it. Any further questions for Mr. Graves? None.
right.
Thank you for your report.
Thank you.
Okay, let's move on to item 12. This is going to be behind tab 9 in your meeting book. The chair recognizes Ms. Doyle to present her appointments to the Scientific Research and Prevention Program Committees.
Members, behind tab 9, you have the list of the 7 Scientific Research and Prevention Program Committee members that I am recommending for approval. And I think We rather cryptically refer to peer reviewers as Scientific Research and Prevention Program Committee members in our statute. Are there any questions?
Any questions regarding these appointments? Hearing none, is there a motion to approve the CEO's 7 appointments to the Scientific Research and Prevention Program committees? All in favor, vote aye. Aye. Any opposed? Motion carries. Now I move to item 13. This is behind tab 10. Recognize Ms. Doyle again to present the presiding officer's new appointments to the Clinical Trials Advisory Committee.
And members, I have 3 advisory committee members that are going to be appointed to the Clinical Trial Advisory Committee. All 3 of these members, I think, are in response to feedback that was given to the Clinical Trial Advisory Committee when they made their presentation in terms of expanding the representation of that advisory committee to include people that were working outside of the areas of Houston and San Antonio and Austin and Dallas. So they've done great work in terms of recruiting these members.
Does anyone have any question regarding these 3 individual appointments to the Clinical Trials Advisory Committee? Very good. Is there a motion to approve the 3 appointments to the Clinical Trials Advisory Committee? All in favor, vote aye.
You're going to introduce me first, so don't share until I introduce me.
And while that's getting straightened, I will add one additional note that is behind tab 10, I believe, for you. The University Advisory Committee is the only committee where you don't take action to approve the members. They are appointed by chancellors of their systems. And so we have included a letter from the Texas Tech University Chancellor appointing Dr. Clegg to the University Advisory Committee on behalf of the Texas Tech University Health Science Center in El Paso.
Very well, thank you. Any additional questions? Let's move on to our advisory committee presentation. This will be the Advisory Committee on Childhood Cancers and will be done via Zoom. Dr. LeBeau, will you please introduce Dr. Richard Gorlick and Dr. Will Parsons to present the Advisory Committee on Childhood Cancers report? And we will just say congratulations to Dr. Parsons on his award that was just approved today.
Thank you. You have met Drs. Gorlick and Parsons last year. Dr. Gorlick is Division Head and Department Chair in the Department of Pediatrics at the University of Texas MD Anderson Cancer Center. He holds the H. Grant Taylor MD, W.W. Sutow MD, and Margaret P. Sullivan MD Distinguished Chair in Pediatrics. He serves as the director of the Pediatric Sarcoma Research Laboratory, which focuses on advancing targeted therapies for childhood cancers and understanding the development and progression of osteosarcoma. And you may recall that last August, I believe, you approved a multi-investigator research award for Dr. Gorlick, which is developing novel therapies for osteosarcoma. Dr. Parsons is a board-certified pediatric oncologist and professor in the Department of Pediatrics at Baylor College of Medicine and the interim director of Texas Children's Cancer and Hematology Center. His work has been instrumental in characterizing the genetic landscapes of a variety of pediatric and adult cancers, including the first identification of IDH1 and IDH2 as critical oncogenes in gliomas. His current research primarily focuses on the clinical application of genomic technologies in pediatric cancer care. Drs. Gorlick and Parsons, thank you for joining us.
Welcome. Good morning.
Thank you so much for that kind introduction, and thank you for the support for our MIRA that was mentioned, as well as it sounds like Dr. Parsons is about to get support as well. So, you know, we apologize for not being in person, but I hope you're starting to see our screen. You know, a lot is going on in Houston, and actually I had planned to divide this presentation with Dr. Parsons, but actually Baylor's Cancer Center is undergoing a site visit by the NIH right now. We had hoped for him to be able to present, but pediatrics is actually being presented at the moment. So he's very sorry that he couldn't be in two places at one time. So I'm going to give the presentation on behalf of our group. Next slide. Sorry. 1 second. Okay. So what I'm going to try to do is present a brief overview of childhood and cancer in Texas, summarize very quickly CPRIT achievements, talk about our ongoing efforts, talk about our strategy, and summarize and provide next steps. So talking about childhood cancer, Texas has one of the highest cancer populations in the United States, largely concordant with its population, meaning it has the second highest population in the United States, and therefore the second largest amount of cancer. In Texas, over 1,800 children per year under the age of 20 are diagnosed with cancer. And if you include the adolescent and young adult population as well, you're talking about an additional 7,800 folks with cancer. The number of children in Texas is projected to increase, and that largely reflects the fact that the population of Texas is growing. And Texas is a very young state where its median age is 35. So with that many children, we expect cancer to grow, and it's important to continue to invest in improving those outcomes. Adolescent and young adult cancer is a particular challenge. It's defined as a group of patients who are 15 to 39, but this is a group that both falls through cracks in medical systems, not fully embraced by pediatric hospitals or adult cancer centers or adult hospitals. And at the same time, where the rates of cancer keep rising, particularly things like colorectal cancer keeps going up in incidence. And, you know, this is something that's picked up by the popular media repetitively. More young people than ever getting colon cancer. Younger folks are getting cancer more often. We don't understand why. But it's certainly a phenomena that's occurring. We want to really clearly send the message that CPRIT has done a lot for pediatrics, and we really want to acknowledge that. We sort of believe it's an area that's worth investing, but CPRIT has invested almost $500 million on research in childhood and adolescent cancers. That's 13% of its entire portfolio. We completely recognize that at the national level, pediatric cancer isn't supported with the same commitment, with generally under 4% of their funding going, so proportionally 3 times greater commitment is provided. This has funded work on particular types of cancer, clinical trials, and has impacted a very large number of patients. The investment in cancer is spread across programs, and that includes academic research, prevention, and product development, with most of it falling in the academic research bucket. Programs that specifically support pediatrics include the programs that recruit childhood cancer researchers. Michael Taylor this year was a very prominent recruitment from Canada. It's a pediatric neurosurgeon who was brought in as a CPRIT established investigator and came to Texas Children's, where, to be honest, he's a great partner for folks throughout Texas. Core facilities, what CPRIT provides is the ability to provide a second award, a second application if it's in the areas of pediatrics or prevention. A number of successful applications exist, and these cores act as an organizing principle for a lot of what we do. Most recently, the MIRA program also allowed an additional application if it was focused on pediatrics. As Dr. LeBeau acknowledged, we were very proud to be a recipient of an award. In the first year, a PEDS award was allowed, and obviously this support is making a huge impact. The impact of the CPRIT funding mechanisms is not only on research on tech in Texas, but really sort of allows Texas to lead the nation. You know, some of the things that have been supported by CPRIT for pediatrics have become national resources. Some of those things include, you know, childhood cancer models, data analysis, data storage. And, you know, we continue to recommend that specific calls are made for pediatric programs in funding mechanisms. Examples of individual research projects that have been tackling big problems and are critically important are some of the proposals listed here. It's always a selection of many proposals, some of which are, you know, we're able to mention, but space doesn't allow us to mention all that are impactful. And Texas is clearly leading the nation, you know, whether it's in precision medicine, preclinical testing, or data, or other things. Investigators that have been supported have made huge differences, and that's really key to the success of these efforts. Next slide. So, a core facility support occurred in 2024. 22 applications were submitted, 9 were funded. MIRA was reinstated. 18 applications were submitted, 5 were funded. And an RFA was created for this particular other mechanism. Part of the reason we need a continued investment in pediatric cancer is progress remains, has been made in many areas, but many areas not so much. Whether you are talking about pediatric patients with brain tumors or metastatic solid tumors, or selected malignancies like bone cancer, which is my focus, there hasn't been as much progress as we would have liked. So what are we doing to try to, you know, continue to make progress? Some is the advisory committee itself serves as an organizing activity for, you know, research in different areas throughout the state of Texas. Again, and some of the groups are even focused on issues like, you know, delivering better care throughout the state, with the state being a huge state. And I really highlight the efforts of the Frontiers Group, which is the name they chose for themselves, which is really about ensuring that the benefits that are accrued by CPRIT accrue to the entire state. We continue to be very inclusive in our membership of the advisory committee. And most recently, we've invited Dr. Amir Mian to join us. He's the head of pediatric oncology now at Dell Children's in Austin. And we continue to engage active pediatric leaders and pediatric oncologists throughout the state to really help define how we approach making things better throughout the state. In the past, we've put a lot of recommendations forward to CPRIT, and some of them are simple things that are like enhancing website and dissemination of information. And we're really proud to share that a lot of these things actually have already been done. So the enhanced website, the addition of MIRAs, the expanded opportunities to cores, they have all sort of occurred, while others are still, you know, being worked on to be implemented in the short term. And again, a lot of our emphasis has been supporting funding mechanisms or suggesting supporting funding mechanisms that accrue patients throughout Texas and benefit the entire state. One of the ways we organize ourselves is through the support of the Carson Leslie Foundation and their dynamic advocate leader, Annette Leslie. Every 2 years, we've had a meeting called the Researchers Roundup. Last year, we were able to talk about the researchers roundup that happened in 2022. We had a meeting once again in 2024 with some of these photos from the researchers roundup, and you can see many familiar faces in the photos. And this serves as our means of sort of organizing how we suggest we improve pediatric oncology care in the state of Texas. Members of the committee convened in Dallas on November 10, 2024. And really what we wanted to do was be more specific in defining an RFA that will address the key issues in the state of Texas for childhood cancer. We had 2 breakout sessions, which were really defined by the prior meeting. And one of them focused on issues of AYA, survivorship, and health disparities, while the other focused on pharmaceutical industry interactions, drug development, product development, health disparities, and clinical trials. And again, I want to highlight that these committees often bridge to other committees in CPRIT that are focused on the same topic, with sharing of membership allowing these things to be coordinated. And with this, we came up with a number of recommendations. The first recommendation is it's really critical to continue doing all the things that are successful thus far, meaning we really encourage the core grants, the MIRAs, the research grants, the recruitment awards. They have and continue to make huge impacts, and please accept our gratitude for them. There are the continued immediate opportunities, things that we'll continue to work on, model sharing, data harmonization, things that with resources that we've already been provided are things we can still do better in terms of sharing, meaning the research has been great at each organization, but we have to share more of what we're doing across sites. And then what we really came to is a suggestion the mirrors are continue to be focused to really take on particular tasks. And some of the tasks that are really important are the ones that have been highlighted in the red boxes. We have to define the population at risk. We must, include implementation scientists. Again, we have to figure out how to make the work we're doing benefit everybody in Texas. And we weren't specific about prescribing, what this work actually has to have as sort of its endpoint. Some of these things can be supportive care endpoints, like, survivorship services or fertility preservation. But we did think the most important thing was actually cancer treatment trials, because survivorship and the amount that survive is really the critical issue for us. With that, I really want to thank the CPRIT leadership. One of the things that is an energizing force for us beyond our membership is really the degree of engagement. They come to all of our meetings. They join our advisory committee. They are an important part of the team. We almost feel like they're as supportive of pediatric oncology as we are, which we're very appreciative for. And really, I want to just extend my appreciation to Texans, the CPRIT Oversight Committee, and CPRIT for really their bold thinking in keeping pediatrics front and center. CPRIT has already led to remarkable innovation and scientific breakthroughs that have benefited the children with cancer in Texas, as well as throughout the country. Obviously, the visionary leadership really helps. And, we hope you're going to continue to support the PEDS initiatives, and please consider other initiatives that will try to expand the benefits that are being made to all of the state. So thank you so much for the opportunity to present on behalf of the committee. If there's time, I think there's an opportunity for questions and answers, but thank you for the opportunity to present.
Thank you so much, Dr. Gorlick, for that excellent presentation. And we echo your commitment to the scourge of childhood cancer. Any questions for Dr. Gorlick from the Oversight Committee? Dr. Rosenfeld.
Thank you. Excellent presentation. Just have a quick question. You mentioned CPRIT's commitment to pediatric oncology at the national level. And I was wondering if you could provide some more color for that. And maybe are there any presentations, say at ASCO or ASH or AACR, where the contribution of CPRIT to pediatric cancer is specifically mentioned?
Yeah, I think we have to find better ways to publish what CPRIT has done. And, although individuals, institutions, and research are often acknowledged, so, whether it's, folks at, MD Anderson or Texas Children's or Southwestern or Texas Tech or any of the other programs, these folks are being featured for the work they do, and it always acknowledges the CPRIT support that's been part of it. I think what's missing is a lecture or a publication that basically shares what CPRIT has done as a collective rather than focus through the eyes of an individual's work. I think one of the things we're thinking of doing as a wrap up to the researchers roundup is really summarizing some of the recommendations we made, but a lot of the intent of the publication would really be sharing with the rest of the nation the fact that CPRIT has been able to drive forward research in pediatric cancer throughout the next nation. It really, Texas in so many areas leads the nation, and this is one of them. And we do need to highlight it more.
Thank you.
Excellent points. Thank you very much. Dr. Rice.
I'm just going to ask a quick, first of all, thank you very much, Dr. Gorlick, for your presentation. Again, great to see you this year. I'm interested in ROI. Maybe that's a surprise to those in the room that have heard me ask questions earlier, but wanted to ask the question that you said the word data harmonization, and I wanted to just sort of pivot on that for a second. I'm assuming either the MIRA or the CORE grant somehow enable investigators to aggregate data in some way, kind of like, so that you have this data harmonization, meaning they can compare their work and assure that the ways that data is organized in some large data set is harmonized, is made so that it can be put into a larger data set and made sense of. I hope you're still there.
Yeah, I am still there. I apologize. There's been
no
some
problem.
camera issues and I've been going back and forth. I apologize for that. Somebody is working on the technical issues, but I'm here and I hear you.
Thank you. So the point I was trying to ask about is that, would you say is it fair to say that, but for the CPRIT grants, the degree of data harmonization that would have otherwise happened would be less? And I'm trying to get to the point that the data harmonization, kind of like the biobank we talked about before, and allows us to to make a gigantic step further than we would have otherwise by sort of combining the research and using that harmonized data to then, have deeper insights. Is that a fair comment?
Absolutely a fair statement. One of the things that CPRIT did that I thought was brilliant when, in fact, MD Anderson received a core facility award and Southwestern did at the same time, is those two projects were related. And what they insisted upon is that the data was shared across the two projects. So the data acquisition was linked to sort of putting the data in a public domain. That worked very effectively, but the problem is each institution has their own sort of platforms, if they're not fused in that way, for how they capture data. And it's a lot of an electronic function to sort of ensure that, we all use a system that allows them to sort of take the data interchangeably. And I think we still can do more in that regard. I think when opportunities exist, there is harmonization. And I don't think in pediatric oncology, it's ever an unwillingness to share the data. It's the fact that they're in two different software programs that don't talk to each other entirely well. And at times there isn't a data dictionary for, let's be honest, some databases code, things differently. So, metastasis is 0 or 1, or it's yes and no. And even though the information is captured, it's not as shareable as it should be.
Thank you. And your example is exactly right. Data dictionaries where one facility might call, again, some kind of a cell, a certain thing, and another calls it another thing, and then those data sets don't match up. but I'm asking you, do you feel like CPRIT has significantly advanced the data harmonization that we would have otherwise achieved without CPRIT?
I do think they've helped, but there's more opportunity.
Okay,
That would
thank
be
you.
the short answer.
Thank you.
Excellent points. Thanks for getting that on the record. Any other questions for Dr. Gorlick? Very well. Thank you for meeting with us today, Dr. Gorlick, and we look to hearing from you again in the future.
Thank you.
Now we're going to move to item 15, tab 11. Sorry. Discuss the final order adopting changes to Chapter 703.
Good morning, members. The final order is behind tab 11 of your meeting books. The final order is to adopt the changes that we presented at the November Oversight Committee meeting. Publish those changes in the Texas Register, and we did not receive any public comments, so there are no changes as we presented them in November. So next steps would be to file with the Secretary of State, and they would become final 20 days after that. And that's it.
Very good. Any questions for Ms. Eckel? Hearing none, members, is there a motion to approve the final order adopting the rule changes to Texas Administrative Code, Chapter 703? All in favor, vote aye. Opposed? Motion carries. Thank you very much, Ms. Eckel. Now we'll move to item 15, which is behind tab 12. The chair recognizes Mr. Grant Weaver to present the Chief Financial Officer's report. Good morning.
Good morning, members, members of the public, CPRIT colleagues. Glad to be here this morning. I'm Grant Weaver, the Chief Financial Officer. Y'all have the full presentation covering the budget and related items in your packet. So admittedly, out of craven self-interest, but also with an eye to you and the full slate of business you've been pouring through this morning, I'm going to attempt to be good. I'm going to attempt to be brief, and I'm going to attempt to be gone and let y'all do more important things. But with that said, in the presentation materials, we provided you a copy of the budget for the period ended November 30th, 2024. You can see our budget has 4 components. Our indirect administrative strategy is about $5 million. Our direct administrative strategy, including a transfer to support critical administrative functions, is about $17.7 million. And those compare to our research program budget of about $245 million, and our prevention program, which we fund at the statutory maximum, of about $27 million. So all in between the 2 administrative strategies, you can see that those come to about $23 million, which is well below 10%. And I think y'all would find that's comparable and reasonable compared to other organizations of our size. Moving on to the revenue report, I wanted to highlight a couple of interesting things for you. As of today, we've earned about $11 million total in revenue sharing since inception. Of that amount, about $4.5 million is attributable to royalties from Wellereg. Our esteemed and distinguished previous Chief Financial Officer has updated y'all about the pace of sales on Wellereg and the increasing international distribution that drug has achieved in the last several years. We've seen that continue. Just earlier this month, Merck published an update to shareholders and the public about its 2024 sales. And in that report, they highlight the fact that year over year, Wellereg, so from December 31st, 2023 to December 31st, 2024, Wellereg saw a sales increase of about 133%. And Merck presents that information in the context of its sort of 15 or so products that they identify as the primary drivers of their overall sales. And Wellereg's growth, again, year over year of 133%, stands head and shoulders above all its other products. The next closest one is about a 30% sales increase year over year. And so I highlight that just to say, as you look at our royalty payments, just as a data point, in October, we received a royalty from Merck for Wellereg of about $205,000. Just at the end of January, we received another one for about $240,000. And then if you look back a year ago to January of 2024, we received about $108,000. So what we're seeing as far as revenue collections from those royalties is mirroring what Merck is telling us in their shareholders report, just that we're seeing very healthy growth in that regard. And certainly with some of the conversations we've had recently, talking about ROI, talking about revenue, return on investment, and so forth, it's very encouraging. And then finally, I would just say, specifically for the fourth quarter in Merck's report, they attribute a lot of the growth in the sale of Wellereg to U.S. sales only, and the increasing indication that they're finding for that drug. And so I say that just to say, even though they have been expanding internationally, it doesn't sound like that has been the primary driver of the increasing revenues that we've seen thus far. And so I'm hoping that's a harbinger of even more good things to come going forward, and that when I come back here in a few months, I'll have more good news to highlight in that respect. You can see we've included some information on our first quarter performance measures that we submitted to the LBB, the governor, and other state leadership. Nothing too remarkable there. You can see for our prevention services, we're right on track, if not a little bit ahead of schedule for our yearly targets. And to date, or I should say for the first quarter, we didn't have any company relocations, but we still got 3 more quarters, obviously, to report throughout the year as we go along. The last topic I wanted to briefly mention is an overview of our debt and our bond issuances. In December, we worked with PPFA to issue just under $70 million to support prevention and research awards from 2019 through 2024. That brings us up to about $155.5 million issued thus far for fiscal year 2025. And in total, about $3.3 billion since the agency's inception. And sorry, just to back up a little bit, when we worked with TPFA in December to issue our last tranche of commercial paper, it was a taxable issuance with about a little less than a 1-month horizon till maturity, and they issued that at a taxable rate of 4.65%. Anybody who's been following the financial markets, even casually recently, could probably tell that's a pretty good rate. And we're blessed with a lot of resources here in the great state and prudent financial management from state leadership. So state of Texas debt carries a AAA rating, the very highest rating you can have for across all 3 of the financial rating houses. And that's even a little better on average than the United States credit ratings. So again, I highlight that just to say we're getting the very best rates we can possibly get when we issue that debt, as evidenced by the recent 4.65%. So with that said, I think I've covered the highlights, but happy to take any questions y'all might have.
Mr. Weaver, thank you for your report. Just to close the loop on the discussion about Wellereg and the royalties and profit sharing payments. For the record, can you just describe to us where that money goes when we receive that from those profit sharing companies?
Yes, I believe it goes back to the state of Texas into an interest and sinking fund that's reapplied to help defray the cost of our issuing that debt.
Very good. Thank you. Any other questions? Very good. Thanks for your report.
Thank you.
Let's move on to item 16. The chair recognizes Ms. Heidi McConnell, our deputy officer, to update us on CPRIT's Innovation 7 conference.
Thank you. I have a very brief report. Mark Loeffler, Justin Moran, Shannon Cusick, and I have a small working group now. We've engaged with Innovation Event Management, which is the meeting conference planner.
that we have on contract. And we are working with them to finalize our venue, conference venue RFP solicitation, and that will go out statewide once we're able to issue it. It will be because of IEM's relationships around the state with various venues. We do anticipate hopefully some options for the next conference that we'll be able to choose. And we've also worked with the programs to kind of narrow timeframes to in the spring of 26 and fall of 26, taking into account some of the national conferences around the country that our researchers here and prevention programs would participate in, so we can kind of take those out of the running for potential dates. But when we have more information, I'll be updating you again.
Thank you very much for that report. Are there any questions or comments regarding the Next Innovations Conference? Very good. Thank you, Ms. McConnell. We'll now turn to item 17, which is behind tab 14 in your meeting book. The chair recognizes Communications Director Mark Leffler to give the update on CPRIT's communication activities.
Good morning, members. Thank you. You'll find my report, as you said, behind the last tab in your booklet. I always enjoy being last because I know I have you right in the palm of my hand. So thank you. I will go fairly quickly. As Kristen mentioned, we did publish the cancer plan in December, so after the last meeting. So you did get a preview of that, I think, at the last meeting. So we were able to publish that in December and get that done, and then move on to annual report. And we're not going to preview that here today, but I hope all of you get a chance to check it out. We're continuing to expand the annual report every year. So this is, I think, the third year I've done it. So we're continuing to grow it. It's got over 3 dozen short articles about what CPRIT is doing and updates from grantees, companies. So I really encourage everyone to check it out if you haven't had a chance already. As Kristen had mentioned, the theme this year was clinical trials. So we had a challenge of trying to present clinical trial information in an easily digestible way. And so we came up with a flip card system where people can see the basic information. If they want more information, they can go to clinicaltrials.gov. So check that out if you get a chance. And also, in addition, to support that theme, we created a new clinical trial primer video featuring Dr. David Gerber, which we're pretty proud of. I think it's a great introduction to clinical trials. So if you ever need to explain that to anybody else, send them to that video because it's just the basics. It's really well done. So so please check that out if you get a chance. Also, the only other thing I'll point out is we have been continuing to try to ramp up our earned media. As you know, we're prohibited from spending any money on marketing or advertising for CPRIT. So everything we do to try to get to raise awareness for the agency is going to be based on earned media. So we're always trying to increase that earned media with interviews, whatever we can do, working with local media to do that. I think since the last time we had met, we were able to get an interview for our CEO, and she did a fantastic job here with KXAN, and it's their local state of Texas weekend report that they do. So it reaches a statewide audience. And we hope to continue more of that to try to get the word out about CPRIT. That's always a topic of discussion. We're open to ideas, but getting what we can through earned media, because we've got a great story to tell. People are very interested when they find out about it, and it's just trying to rise above the noise of media coverage, and that's what we're trying to do. So with that, I'd be happy to answer any questions.
Thank you very much. I just want to give a shout out to you and really the whole CPRIT team on the work in the last few months with the cancer plan and the annual report. It's very well done. I've been on the website and looked at it. I agree with you. The video with Dr. Gerber is one to keep in your back pocket, especially it's easily disseminated and it's language that, you know, patients can understand. I thought it was very well done as well.
Thank you.
Any other questions, comments? Very good. Thanks for your report and all your work.
Thank you.
Okay, now we're moving to page 20 in our meeting book. This is now agenda items 18 and 21. The Oversight Committee will move into closed session to consult with CPRIT's counsel. For members of the public watching this meeting, we have reached the end of our agenda. I do not expect to act on other matters after returning from closed session before adjourning the meeting. We go into closed session pursuant to Texas Government Code 551.071 to consult with General Counsel. The following CPRIT staff shall join the Oversight Committee in closed session: Ms. Doyle, Mr. Ellis, Ms. Eckel, Ms. McConnell, Mr. Weaver, Mr. Burgess, and Dr. LeBeau. The current time is 10:49 a.m. The time is 11:41 a.m. Items 19 and 20 will not be taken up. Item 22, we will not take up standing items 19 or 20, but turning to item 22, the next regular Oversight Committee meeting will occur on May 21st, 2025. Before we adjourn, I'd like to discuss a topic for a future board retreat that has been floated for some time. The Special Issues Subcommittee has met and started to come up with options for a topic discussion for a board retreat that would likely occur prior to our next scheduled Oversight Committee meeting. The meeting would last no more than 4 hours, and we would have some time to address likely 2 policy issues and potentially 1 process issue. If there's any recommendations or additional thoughts on that, please get with Ms. Doyle or myself. The Special Issues Subcommittee will continue to meet to firm up our agenda for that board retreat. If there are not any further questions, the chair moves to adjourn this meeting. Is there a second? All in favor?
Aye.
Motion carries. The meeting stands adjourned at 11:42 a.m.